All right, so my stomach is currently growling, as I'm sure many of yours are, so we'll try to get through this and get everyone off to lunch. Thank you for sticking around. So I'm a postdoc at WashU in the Dozenbach lab, and I'll be talking about precision mapping of the human subcortex. All right, so I just want to start out by giving a huge shout out to the co-first author of this work, Deanna Green. You can see there with her there with her thumb up. She's giving the thumbs up for the coding that I'm doing as she is relaxing on my chair. And also to Nico Dozenbach, who's my mentor, and Deanna Barch and Joan Luby, who are funding me on a T32 to do all of this work. And so this is the title of the actual paper. It's currently in revision. And I also want to thank all of those co-authors. All right, so I'm just going to start off with some clinical significance of the basal ganglia and thalamus. So both of these regions are treatment targets for multiple neuropsychiatric and neurological impairments with deep brain stimulation, specifically the ventral intermediate nucleus of the thalamus for essential tremor and the globus pallidus for dystonia. However, success rates vary across stimulation site. And one of the things that we're broadly interested in Nico's lab, especially long term, is how can resting state fMRI be leveraged to inform treatment targets? And to this extent, one tool that we want to use is this idea of precision mapping, which is simply the acquisition of large quantities of data in individuals, which enables the reliable estimate of individual functional brain organization. So in each of these graphs here, you have reliability of functional connectivity data as a function of time. The panel on the left is in the ABCD data set. You can see as you go from 5 to 10 to 15 to 20 minutes worth of data, you boost reliability within subjects. And then once you get out to about 45 to 60 minutes worth of data, you can achieve good to excellent reliability in individuals. So the low reliability at small data quantities can be overcome by just collecting more data. So what have we learned so far from precision mapping? The first thing is that functional organization shares common features across individuals, but it displays measurable individual specificity. So I have an example here on the right from Evan Gordon's 2017 paper, which was the original Midnight Scan Club paper, where you can see in black here there's variants of the salience network that exists in the medial prefrontal cortex, but they exist in anatomically variable regions of it. Now, if you just look at the average, you see that there is no salience network represented. It's all default mode. So group average studies necessarily blur this phenomenon. There's a really wonderful paper by Katerina Graton that I encourage everyone to read, where basically what she's doing is quantifying the variance in functional connectivity that's shared across individuals and variance that is unique to an individual. And one of the main findings from that paper was about half of the variance in functional connectivity is individual specific. So again, group studies would be missing out on this. However, when you're engaged in a task or when you look at a subject session to session, there's not much variance that's accounted for. And this is really suggesting that functional connectivity is more about the individual than what they are currently doing. And I think that this is extremely important for diagnostic purposes. And then lastly, we recently published a paper doing precision mapping in the cerebellum. And we found that one of the findings was that the frontoparietal network is actually overrepresented in the cerebellum compared to cortex, and that's the case across all subjects. So I'm just pivoting slightly just to give a little bit of background about what we know about subcortical organization. We know that there's integration of distinct prefrontal cortical regions in the basal ganglia, and we also know that there's structural and functional integration of both frontal and parietal networks in the basal ganglia and the thalamus. So the purpose of this current study was to delineate network-specific and integrative functional network organization within individuals, and I'll tease that apart in a little bit. So the first question we can ask is, what is the functional neuroanatomy of the subcortex? How are these functional networks laid out? And so in this panel, it's the group average from the MSC. And you can see broadly that motor networks are represented in the ventrolateral thalamus. So this is somatomotor hand, motor face, as well as the cingulopericular network. And this is a theme that we'll keep coming back to. Then other networks like the frontoparietal network are located in the dorsal thalamus as well as in the caudate. And the default mode, which everyone knows and loves, is located in the medial thalamus as well as the caudate. One of the really cool and powerful things that you can do with reliable individual subject data is you can start to look at variability in how these networks are laid out from individual to individual. So you can see here that the two motor networks in the cingio-opercular network have high subject overlap. In some cases, all subjects overlap in the same exact anatomical location. But then other networks like the frontoparietal network, the salience network, dorsal attention, ventral attention networks are much more individually variable. So then what we did was we did a modified winner-take-all analysis where we allow voxels to either be network specific, which means that there is one functional network that is represented in that voxel, or multiple functional networks, and this is what we refer to as integration. So areas of the subcortex in which one network is represented are colored by these colored-in squares here, and where you see stripes in the images are where there's network integration. And then one other point is that organization exhibits both common and individual specific features, just like in the cortex and cerebellum. So you have the example here in MSCO2, in the posterior putamen where the cingio-opercular network is represented. However, in another subject, MSCO6, the somatomotor hand network is represented. So what we wanted to do is we wanted to categorize voxels along two axes. One was integrative versus network specific, and the other one is group versus individual, which means common across people or individual specific. So you'll see in the next slide is that each voxel in the subcortex will be categorized into one of these four categories. And this is what the breakdown looks like. So in blue is where there is one network represented, and it's that same network across most individuals. In green is where there is one network, but it tends to be variable as to which network is represented in that location. In yellow is where there's integration of multiple networks, and it's the same networks across all subjects. And then in red is where there are multiple networks that are integrating, but they tend to vary slightly from person to person. And then what we can do is we can quantify each type of functional zone. And you can see that not the majority, but a lot of the voxels are network specific and common across people. But the category that I really want to focus on going forward is this group integrative piece here, where there are multiple networks that are integrating and they're common across individuals. So we found three such group level integration zones. So I'm just showing the figures here. This was all quantified using cluster analyses. But the first zone that we found was a cognitive integration zone in the caudate. And in this region, there was integration of the default mode, ventral attention, frontoparietal, and salience networks. Located around the pulvinar of the thalamus, there was a visual attention integration zone that integrated the dorsal attention network and visual network. And then the one that we found very interesting was in the ventral intermediate nucleus of the thalamus, where you have integration of both motor networks and the cingio-opercular network. So for the remainder of the talk, I'm just going to be focused on that motor integration zone. So what we can do is we can put a seed in the ventral intermediate nucleus of the thalamus in this yellow area right here, and we can just see what the topography of correlations are on the cortex. So this is in just two example subjects, MSCO1 and MSCO2. On their cortex are the network boundaries for, say, the cingio-opercular network in purple, in motor face in orange, and in motor hand in turquoise. And you can see how nicely the correlation structure maps onto the individual specific network boundaries for these two subjects. And then we can quantify the magnitude of the correlations of what we're calling integrative, and you can see very clearly that these three networks are represented and no other networks, and this was the case for every individual. And this has, we think, a lot of clinical significance. So this spot, this exact coordinate in the ventral intermediate nucleus of the thalamus is the stimulation site, the DBS stimulation site for essential tremor. And the really interesting part about this is that DBS for essential tremor is about 99% effective. It works across almost all people. And it's located in that integration zone where everybody shares the same three networks. Conversely, DBS stimulation in the globus pallidus for Parkinson's disease is an individually variable piece of the globus pallidus functionally. And this site is only about 30 to 50% effective. And so we think that if these individuals are precision mapped and we know which network to target in the globus pallidus, the hypothesis would be that you would boost efficacy rates. So in conclusion, the basal ganglion thalamus contain integrative zones shared across individuals. We noted reliable individual variability, and we think that differences in success rates of DBS may be due to individual variability in functional organization. And as such, precision mapping may be a powerful tool for increasing DBS success rates. So that is it.